Okay, hi you guys. Let's go ahead and I'm going to share my screen with you. Okay, so this is the optional lab video right here. And we're going to, sorry, let me find the page. We're going to talk about a little bit more about just different bioinformatic tools that you can use to just understand a little bit more about a certain gene of interest, right? So here, I'm going to use GAPDH. GAPDH is what's called a housekeeping gene. That means that it's a gene that is pretty ubiquitously expressed. That means it's expressed pretty much in all cells, is constantly expressed. And so when you do different type of um, experiments, a lot of times you have like a standard or like some kind of housekeeping that you can measure to. So similar to standard, if you were to like run, for example, a Western blot, and you kind of want to know, is this amount of protein that you're getting on this Western blot, is it a lot or a little? It's kind of like a lot or little relative to what. So a lot of times you will compare it to a control standard. And so GAPDH is one of those ones that that you tend to use. So I'm just giving you a little bit of background so you know why I kind of picked that. So because it's a housekeeping gene, I expect it to be expressed in pretty much every cell, no matter which cell that I did my extraction from. I expect it to be expressed in every cell, and I expect it to be expressed in pretty uniform level, probably across all the cells. And I expect it to be also pretty conserved, just because it's so ubiquitously expressed in human. Most likely is conserved because it probably has a um, pretty useful function if it's expressed everywhere, right? Like you can imagine, like why would the cell take the time to express a gene that it or a protein that it doesn't need? So it's probably pretty conserved, and I could probably find it in other species. So what we're going to do in this optional lab is we're just going to use some other bioinformatic tools. I kind of just want to introduce you to some other bioinformatic tools. This is going to make a lot more sense when we actually start doing the project that we're going to do at the end of the semester. And I'll probably introduce that project in another week or two or so. That we'll introduce the project so you guys have an idea of like why am I making you guys do all this blast stuff? Okay, so here is the instruction. So the lab comparing DNA, in case you haven't opened it yet, this is pretty similar to what we did before, but it's going to talk about evolutionary wise, like how you understand it. So real brief, let me kind of go through it. So this is what's called a cladogram. So the way that you would interpret this is that is basically using this graph to kind of figure out how close things are to one another. So based on this graph, these two different organisms are probably a lot closer to one another compared to this one right here, right? I actually don't know much about, I think these are like insects or something. I don't know too much about these different species, so I can't speak more than that. But by looking at this, I can tell you which species is close to one another, right? So evolutionary wise, we use DNA sequence a lot of time, which is kind of what we're looking at today, to be able to kind of tell how close things are from one another. We can actually use it to kind of figure out when certain things evolve. So here in this picture right here, being able to kind of map the sequences and how close they are evolutionary wise, I can tell you that the jaws was probably developed first, right? And then over time, we developed lungs. Probably when we started being able to move from water to land, we developed lungs, and we were able to then evolve from the lungs to having these other components of like dry skin and hair and tail. So this kind of using sequences, we can actually see the evolutions of different genes that start coming up to give us certain phenotypes, right? So that's kind of what we're looking at here. Is this cladogram kind of shows you the progression of evolution. Okay, so what we're doing, the reason why I pulled this one online, this is the information, this is talking more about this, is I actually wanted to go down to this part. So in this project, they were actually using it to try to find evolutionary. We're not really looking at it for evolutionary so much right now. We're just trying to explore things, but I wanted to put this on here so that you would have these screen grabs that kind of show you how they went about it. So they're using BLAST here. So here they were given a number of gene files already. So they already knew what genes they wanted to put in here. So similar to what we did last time, you're taking a gene, you're going to search the nucleotide bases, and then they were able to stick it in there and kind of be able to do a search. So here, this is where your FASTA would go. Remember that FASTA is those DNA sequences that you get from your search. So you were able to get the genes, and then from the gene you can so last time for the last lab, I didn't care if you guys did protein or if you did mRNA or if you did nucleotide. It was more of just getting an idea of what your sequences are. But ideally, you would actually get the DNA. And the reason why is because of all the different isoforms of mRNA. And plus, like once you have the isoform mRNA and you try to put it in BLAST, it's going to be very stringent because it's only going to look for other mRNA that matches exactly. So you're not even going to be able to pick up on all those other isoforms. So we want the raw DNA because it's going to have all the different possible exon and intron segments that you don't see if you pick an mRNA. Because so recall that the mRNA is the mature RNA. So at this point, you had transcribed your DNA into RNA, and then the reason why it's mature now is because the spliceosome came in and spliced out. Remember, I said that there's certain parts that's non-coding. It spliced out all the intron, but it can also splice out certain exon that is not present in a particular isoform. So you're removing a lot of information if you just use the mRNA. So hopefully in this project, you will actually use the DNA to get that information. So you're going to put the DNA in here. You can actually pick like which species you want it to be in. You can actually pick how stringent you want it to be. We want highly conserved. If you pick the ones below where it's less conserved, you're just going to get a lot more hits because right, you're not being as stringent. Because remember, it's going to go through the database and it's going to kind of pick the ones that are the closest to it. So picking a little more conserved means that you're more likely to get hits that are close to what you're entering. So you're just going to go through there and you press it in. Remember, this is when I show you guys the different alignments. So this is where your isoforms are. So this is telling you that this right here for your query is about 5,500-ish. So 5.5 kilobase. And this first strand is like the whole thing, this whole mRNA. But you can see that this one right here, the second one and third one, you have a different isoform, right? Because it has this segment, but then there's a bunch of segment right here where it blank, where it did not match. So it didn't have those segment in that particular mature mRNA. And then this one, the fourth one, and fifth one doesn't have it at all. So you can see this is where these different representation just shows you how. So your query is this one. This is what you originally put in. And then it's returning for you all the one that matches that query. So here are all the ones that matches, and it's telling you like how they align, how they match up. So the red is where they actually match up exactly with your query. The blank parts is the parts that they isn't, and that's kind of an indicator of probably where the splice zone probably came in and cut those parts out. So below here, it gives you a little bit more information on like the sequence and like how similar they are to the gene that you're interested in. So it's kind of nice to have this information. This is another image of the alignment right here. This is another one of the tabs. I think is the tab that says sequence alignment. I can't remember. I don't recall off the top of my head. But you remember there's like 4 different tabs. So this is one of the tabs here. And it's actually giving you some information about that particular query versus the sequence. And so it'll tell you like which species it aligns to and where it's found. And it's actually giving you some information on what percentage it matched with. And then it's giving you what sequence so you can actually see like where the alignment didn't happen. So we can see here it didn't happen. Here there was a misalignment. Here there's like some, there's some spacing. So it's not going to match exactly, even if you're comparing two, right? Because different people, for example, we all have like the same genes, but we're not going to have the exact identical genes. Our sequences are going to vary a little bit. So if I was the query and you were the sequence that matches closest, we're not going to be exactly identical. And that might be where you get these like different one sequence. There's also something called SNP, which we'll talk about later on, which is single nucleotide polymorphism. This means that there's only one single nucleotide that might vary, and it's usually very particular location. A lot of times we use this as marker to determine if someone's likely to have to develop like a tumor or certain disease. There's certain marks that are more predominant if you're likely to have a certain genetic disposition, and they're called SNP, SNP, my goodness, SNP. But we'll talk more about that a little bit later when we actually start talking a little bit more about transcription stuff. Okay, so this is the results. And so I kind of want to just give you this site because it kind of walks through and kind of actually also provides all the different, what's happening here? Why did this, sorry, all of a sudden, oh, it split my screen. I don't know why I just did that. What did I do to make it split my screen? Oh, oh my gosh, it's because I'm not on a separate page. Sorry, that's just me. I think I just had a brain fart. Okay. Anyway, okay, let me exit out of this and go back to this original page. Thank goodness. Okay, sorry, guys. Okay, so let's go. So we didn't really search this last time. UCSC genome browser is actually great. It's also a little chaotic, which is why I use it, but then sometimes I don't use it and I go straight to BLAST, and you'll see why. There's a lot of information there, but it's a little chaotic. So let's go figure out where the genome location is. So here is the BLAST. This up here, this is the assembly. This has to do with kind of like a version. So this is version HG38, the most recent version, because as we update our human genome reference, remember I told you that we have a reference genome that's mainly made of like white males, and I've kind of talked about how, that there's certain information that might be in other groups that aren't present here. So over time, we always are trying to update it. So as they update the human genome, they're going to update the version as well. So this is kind of telling you. So, and the versions will matter in that if you're trying to do alignment, there are some small shifts. So if you're using the reference genome 38, but the sequence that you got was, let's say using 19, they're not going to match up exactly with regards to the chromosome location. And so that might give incorrect results because of that. So for right now, what we're doing, it doesn't matter as much, but just keep that in mind. That's what the HG38 stands for. Okay, so we're going to do GAP-DH. See, it already remembers that I looked for it last time. Okay, so here is the genome browser. So my first line right here is telling me here's the gene that I'm interested in. And this is the exon, this is the intron right here. This is the exon, so on. So if I was to ask you like where the mature mRNA is, you would only be interested in gluing these guys together. You wouldn't be interested in gluing. So this is actually all of them, that first one, but this is the example. Here's one version, here's another version, here's another version. So these are all three different isoform of this GAP-DH right here. So if I was to ask you, you would actually just piece these guys together and ignore all the sequence in between. So in your mature mRNA, remember that certain sections of it is spliced out, which when I say splice out means that it's actually removed from it. Like we're not going to include it in our final mRNA sequence. Okay, so I think the first question had asked us where the location is. Okay, let's look at the location. Hide this. Okay, so the location is right here. It's on chromosome 12. And this is the range that's in is 3,855 base pair. You can actually zoom in and zoom out. I'm going to zoom in and zoom out just so you can see. So if I was really interested in this guy, for example, I could zoom in. And this will actually tell me the, so this looks like the protein sequence here. So it's actually telling me the protein sequence. I can also zoom back out. And we'll zoom out again. So this gives us our information. But the original one that popped in, that was the whole site. So if I wanted to, I can actually look over this guy, GAP-DH, and I can also get the genome location right here. So all I did was click on the gene, and it tells me where my chromosome is, which nucleotide sequence is, like where it is. So I would say chromosome 6, and 6,534,000, yada, yada, yada. Here is how big it is. And it actually tells me how many exons there are. And it also tells me which strand is on, if it's on the sense strand or the antisense strand. So this has to do, remember that DNA has directionality. So this has to do with the directionality of the DNA strand, right? So if it's on the sense strand, it's going 5' to 3'. If it's on the antisense strand, is the complementary strand, and it's the one that's going from 3' to 5'. Okay, so I think, let's see. So that's the known location, the multiple isoform of the gene and whether you see alternative splicing. We had already looked at that, but let's look at it again real quick. So let's exit out of this. These are the different isoforms. So these, remember that these are the reference to tell you which one you're looking at, but you can see that this guy has multiple isoform, right? It has this version, and they're all slightly a little different because different isoforms can have different starting sites. They can have different exons that are included, different number of exons as well. These guys all have relatively similar number of exons, but this is how you know if it has a the isoform, because you will actually see it here. So these guys, this does have different isoform, but they're not that different from one another. Sometimes you'll see they actually are pretty different, and the different isoforms can actually have totally different functions from one another, even though they're technically the same gene loci, they can actually have different functions. Okay, so what is the function of the gene in human of the protein that's produced from this gene? So we can get that information by, again, clicking right here. This actually has tons of information for you. And this will actually tell you, where is it? Is it not right here? Typically, it describes it right here. Okay, we'll find another one that tells us that information. Oh, I think because I grabbed, yeah, hold on. I think I might have grabbed it's because I grabbed one of the isoforms and that maybe there's just not any information on there. And the reason why I was able to tell that is that DT that you might have saw me like hesitating for a minute. Anyway, here's the gap deviation. I'm like, I swear it has it in here. So this gives you a description that it stands for glyceraldehyde 3-phosphate dehydrogenase. I always enunciate too fast, in which case my tongue just goes blah, blah, blah. Anyway, is remember I told you the different isoform are the different transcript variants. So transcript is the actual RNA transcript. That's usually what it stands for. And so it's telling you that it also has whether or not it's non-coding or coding. And you should recognize this. This is one of the enzymes that we actually had seen in our we'll see more, but we actually saw it during the whole respiration. So this tells you that this gene has been done identifying moonlighting. So it has moonlighting means that it has like a bunch of different functions. That's what they mean by that. So it has a bunch of different functions. But here, catalyze an important energy yielding step in carbohydrate metabolism. That sounds familiar. Do you guys remember where we did carbohydrate metabolism? It's probably the last chapter we just did where we looked at the glycolysis. So go back and see if you can find the GAPDH in there. But it gives you a little bit more information about it. It tells you, yeah, it's just usually if whatever they know about the gene, they'll give you a little summary. Let's kind of scroll down, see what else it tells us. So it will actually link. These are all different websites that will tell you more information. So this will actually be useful later on for the project. Oops, sorry, slippery hands. We'll tell you more information when we actually do our project. So for the project, I'm going to give you, you're going to work in groups. I'm going to give you a gene sequence. I'm going to give you a certain illness or disease or something that is associated with this gene, and you're going to try to find information about it. So all of these different bioinformatic tools will probably be useful then. So Ensemble has to do, I'm going to go through a few of them that I use a lot. I use Ensemble a lot. It has a lot to do with protein, giving you information about the proteins. GeneCard is great if you don't know that much about gene and you want more fine information. You would click on GeneCard. Another one I use, this is more for the protein if you're interested in more about the protein cell. If you are interested in, I think this gives you the like different GOs, so what else it interacts with. This right here is PubMed, which is where you can find peer reviews. So this would be great if you want to look up peer reviews about this this particular gene and see what can happen. So one of the things that you might want to find out when you do your project, for example, is figure out where the sequence come from, like which gene, and then put the gene and probably the disease that you're interested in and stick that into PubMed, and it'll give you some paper that tells you like what research has been recently published about those with those components. So if you want to design primers, we're not going to design primers in this class, unfortunate, but go back to about how we were talking about how we can use restriction enzyme to do kind of different digest and stuff like that. This is where you could produce the primer. Or if you want to amplify stuff, like if you want to amplify, let's say you have that piece of gene and you really want to amplify it in order to use it downstream for like cloning or whatever, you can use the primer design to isolate it out of the human genome. So let's say you took a sample from your blood. Let's say you did a blood. You gave your blood to the facility and they're going to try to see whether or not you have a particular gene in your genome. You would use a primer in that sense. You would use the primers to target what gene you want to amplify, and a primer would then bind to it and amplify. We're going to talk about PCR later on in the lab, but we just won't be doing the primer design itself. But that's what primer design is. This is a disease that's associated if there's any type of like mutations, or if there's any type of like variation, SNP variation of it. Oh, here we go. Let's look at this. Can't remember if I asked you guys to look at gene express. So is your gene expressed in all tissue and are all the same level? What does this information tell you? So you can go to GText, which I will show you in a little bit, but this is actually pulled from GText. So GText itself is an actual website. Let's just type it in. So here is the GText portal, and I can actually put my gene that I'm interested in, GAPDH. Gosh, I can't type. Oh, it has multiple ones. We just want this guy. So it's going to IG browser. This is to browse the gene itself. This is, again, about the protein. This is UCSC browser. So well, let's click on a different one, IGB browser. So IGB browser is when you want to just see the gene. In general, this is if I want to see like where, this is just to be more selective. I'm not going to apply any of these. This is like if I really was interested in this, in just like, let's say the brain, and so maybe I'll pick it from here. But I'm just not going to pick anything. So here is our gene, and it should have, let's see, not there. Trying to figure out, remember where it shows the gene expression. Oh, it's just right down here. My gosh. There's so many websites, there's so much information out there. You just have to find it. This tells you basically the expression level. So whenever we say expression level in science, we're tending to talk about how much are steady. I'm going to say steady RNA because my PI in my last lab, we actually studied transcription, and we studied nascent transcription, which is stuff that's recently transcribed due to perturbation. So let's say I hit you with a drug. I want to see what genes are being transcribed right then and there because of that drug, which means that within 45 minutes of you being exposed to drugs, how does your genome change? So that's what we call expression in that lab. This is not pertinent to you guys. I'm just giving you some back history of why I say steady state. Steady state RNA is all the RNA that was recently transcribed plus all the RNA that's still in your body. So your RNA constantly gets made, but also gets degraded. So steady state has to do with, at that time, all the RNA that was present in that cell. So anyway, that's what this is. So this is all the RNA that was expressed in that cell at the time and how much. So you can see it's pretty steady. It's pretty steady throughout. But apparently muscle has a big amount, and EBV transformed lymphocyte. These are white blood cells that was induced. So maybe it was induced by a virus to become immortal. Maybe that's why they have more. I don't know why these ones have more. Culture fibroblast. Fibroblast is basically your skin. So maybe it's just growing a lot. I'm not sure why these guys are a little bigger than the other ones, but this is what RNA expression is. So when I ask this question right here, are they expressed at the same level across all tissue? I would say for my, if I was to answer this, I would say, yeah, it's pretty expressed pretty consistently, right? See, and I think the last one, so you can get the protein, you can get the function by clicking on that one link on there on the UCSC browser, right? You can just, that's where we got, that's how we got brought this up and has a function. And we can click up on each one individually and pull up its information for function. The last question is, how well is it conserved? So if you scroll down here, this has to do with expression level. I won't go too much into this, but remember, I'll just explain it so we know kind of what everything is. This is the allele, this is expression level again. So this is just giving you expression level. This is just different markers, like the regulatory elements and markers. So this tells you how well it's regulated. This has to do with histone markers. Histones are the protein that your DNA is going to wrap around. So it's going to wrap around, and your histone, depending which histones wrap around, will give you information on whether or not it is being transcribed at that time or not. So there are markers that we often use to kind of guess whether or not this gene gets transcribed a lot. So here they're telling you that the H3K27 acetylase is marking near regulatory element. So we won't go too much more into that, but just know that the histone, that when you see the H3K27 or H3K3 or whatever, methyl 3, those has to do with which histones your DNA is wrapping around. So let's go down to the part we're actually interested in. So this is the conservatory. So this tells you how well it's conserved. You can see that it kind of lines up with your exon right here, right? So this lining up, you can kind of see it lines up with the exon. And it's kind of telling you how much expression you're kind of seeing. These are all the other animals and organism that you're seeing the genes in. So you don't really see it in zebrafish or whatever extrapolis is. But if you click on this, we can actually see that this is the danogram that kind of tells you what else are possibly expressing the GAPDH and just more information. So I would say based on just how well defined these peaks are and just kind of how much information we have here, that's probably pretty conserved across just evolutionary wise. So there's probably a lot of other organisms that are semi-related to humans that it is conserved in. This is the common SNP. So this just tells you, remember I was talking about the different polymorphism. This is just telling you where they might be located. We'll look at this a little bit more later on, like what SNP is and why we would use it. And then this right here is just all of the other options that you have. These are all different bioinformatic tools, and you can see most of them are hidden. They're just hidden by default. I actually didn't hide them. There's a lot of information that you can get out from this website, which is why I find it a little chaotic. And you can always click on any of these. I think you have to right click. I can't remember. Let me see. I'm on a Mac and like sometimes right click is different. So yeah, I press the control click and I can actually hide it. I can squish it. So let's say I wasn't really interested in the different isoform. I can squish it. I even squish it. So I just move it up. And it just makes it so that I can actually, you would do that basically if you wanted to like see more of the stuff. So if you wanted to, let's say you're interested in looking at CRISPR targets because you're, for those, if you don't know, CRISPR is what you can use to actually do some genetic modification is a way of using, again, bacteria as a tool. They find the Cas9 and the Cas9 was able to come in and like cut certain sequence or recognize this certain sequence that it can then cut, and so that we can actually do genetic modification. So if you wanted to, if you had a bunch of, typically we do more in cells. It's hard to, you can't really do CRISPR on yourself because you can't inherit CRISPR, right? So only the cell that you CRISPR and the cells that derive from that is going to have that modification, so there wouldn't be a point of CRISPRing yourself. But anyway, that's what this would be. So I'm just gonna open it so we can see what it looks like. So if you did that, you can refresh it. And now you can see all the different targets, right? So this just allows you to pick, and these are all different tools. And I think if you click on, let's see where the CRISPR was. I think if you click on this, it takes you to how to go about finding that site. So it's just more information on it. And I think it'll take you to the original tool, which I don't know where it is. Maybe this one is in a separate tool. I can't remember. There's so many information. I always get a little bit overwhelmed when I start doing this stuff. That's why I was hoping I gave you guys clear things to look up, because it can be super overwhelming and you can go down weird, really long, derailing and rabbit hole if you start clicking too much around. Okay, so I hope that kind of gave you guys a little bit more information about different tools. There's a bunch of them. I forgot to tell you guys about this one. This is another one that you can actually look up a particular gene. Yeah, H. And it can give you a little bit more information. So I gave you UC Santa Cruz, but this is also another one where you can actually click on it and it will give you some more information. So this is got the H, we're interested in that one. Tells you the summary of it and where it's found. It tells you where it's mainly found in. So prototype is mammalian primate, gives you the location and information on there. Yeah, so it's just, there's tons of, a lot of repetitive because a lot of them feed off of one another. But hope this helped. And I hope that you guys find all this information interesting and you can kind of see how it could be applied to so many different sector of biology. Okay, I will see you guys. I'm going to exit out of screen share. I hid my thing, so I can't exit out of it. Okay, I'm going to stop my share and I will see you guys in class. All right, bye.